So, what will happen when we put an animal cell into a solution of sugar or salt? If the medium surrounding the cell has a higher concentration of water than the cell, meaning that the outside solution is very dilute, it has a higher concentration of water. So, what will happen? Water will flow from a region of higher concentration to a region of lower concentration. So, naturally, more amount of water will be entering into the cell. As a result of which, the cell will start to swell or bulge. Now, water molecules, they are free to pass across the cell membrane in both the directions. That is, they can move both inside and outside. But more water will be coming inside the cell because the outer solution is dilute. Because the outer solution has more amount of water, more water will be entering into the cell, and this will result in swelling up of the cell. Such a solution is said to be a hypotonic solution. Now, what will happen if the medium or the outer solution has exactly the same water concentration as the cell? Such a solution is said to be isotonic, and movement of water will be taking place inside and to the outside in the same rate. As a result of which, the size of the cell will remain the same. That is, the amount of water that is entering into the cell will be equal to the amount of water that is going outside. So, the cell size will be remaining the same. Now, what if the medium has a lower concentration of water than the cell? So, concentration of water is lesser outside the cell. So, what will happen? Naturally, water will move from inside the cell to the outside. The cell will lose a loss of water. The cell will lose a lot of water by the process of osmosis. Now, such a solution is said to be a hypotonic solution. And in a hypertonic solution, what will happen? Lot of water will be lost into the solution, as a result of which the cell will start to shrink. The cell will shrink because the concentration of water is higher inside the cell. So, water will move from inside the cell to the outside, and the cell will start to shrink. Now, osmosis is a special case of diffusion occurring through a selectively permeable membrane. And you know that in animal cells there is only a plasma membrane. What about plant cells? Do they react the same way when they are put in a hypertonic, hypotonic or an isotonic solution? Now, before studying what will happen to a plant cell when it is put into a sugar or salt solution, let us learn about the difference between plant cell and animal cell. Now, last year itself we have studied about the differences. First, what are the similarities between plant cell and animal cell? Both of them have cytoplasm, cell organelles like mitochondria, endoplasmic reticulum, nucleus, etc. And both of them have a cell membrane or a plasma membrane. What are the differences? A plant cell will have a large central vacuole, while in animal cells the vacuoles are smaller. Plant cells have plastids, and when these plastids have a green colored pigment that is called as the chlorophyll, they are called as the chloroplast. The most important difference is the plant cells have a cell wall. Now, what is the difference between a cell wall and a plasma membrane? The plasma membrane is present in all the cells. Animal cells and plant cells, all of them have a plasma membrane. The cell wall is present only in bacteria, fungi and plant cells. The plasma membrane, as we studied in the last class, is made of lipids and proteins. The cell wall is made up of cellulose. Cellulose is a complex substance, and it provides strength to the plants. The cell wall is made up of cellulose and it provides strength to the plant cells. The plasma membrane, as we already learned, is semi-permeable or selectively permeable. It will allow only substances that are needed by the cell inside and outside. While the cell wall is permeable, it is completely permeable. It will allow all the substances to enter into the cell or go outside of it. Now the plasma membrane is flexible, it is bendable, and the cell wall is rigid. Now the cell wall, it provides protection to the plant cells. So what will happen to a plant cell when it is placed in hypotonic, hypertonic or isotonic solution? What is a hypotonic solution? The surrounding medium will have a higher water concentration than inside the cell, as a result of which water will be entering into the cell. And what will happen in an animal cell? As more and more water enters into the animal cell will start to bulge or swell. And if again more water is entering into the cell, finally the animal cell will burst. But in the case of a plant cell, there is an external covering or a cell wall. And as the cell starts to bulge or swell, the cell wall will exert a pressure against the cell, as a result of which it will protect the cell up to an extent from bursting. But if more and more water is entering into the cell, then the plant cell will also burst. So what happens in an isotonic solution? In isotonic solution, the surrounding medium has the same water concentration as that of inside the cell. So water will be flowing inside and moving outside in the same rate. And net effect is similar to that of animal cell, that is both of them will retain their size. There won't be any changes. And what happens in a hypertonic solution? When a plant cell is placed in a hypertonic solution, as you know, the surrounding medium has a lesser water concentration, so more water will be moving outside from the inside the cell. When more and more water moves to the outer surface, what happens? The plant cell will lose water through osmosis and there is a shrinkage. That is, the cell will start to contract away from the cell wall and it will be seen in the center like this. Now this is called as Plasmo Lysis. What is it called as? It is called as Plasmo Lysis. Now let us do a quick recap of what we studied. What will happen when a plant cell and an animal cell is put in a hypotonic solution? Hypotonic solution means outer solution will be having a higher concentration of water, so water will enter into the cell. In the case of animal cell, what will happen is as more and more water gets into the cell will start to swell and bulge and finally it will burst. But in the case of plant cell, the cell wall is very strong. It will provide a rigidity and a strength to the cell so that the cell will swell. But the cell wall will exert an equal pressure against the swollen cell so that these cells can withstand greater changes. But if more and more water is again entering into the cell, the plant cell will also burst. But up to a certain extent, they are capable of withstanding the pressure. Now in an isotonic solution, water will be entering both inside and coming outside. So net effect will be that the cell will remain the same size. There will be no difference in shape. Now what happens in a hypertonic solution? In the case of an animal cell, it will start to shrink and shrivel up. And in the case of plant cell, plasmolysis will occur. What is plasmolysis? Shrinkage or contraction of the contents of the cell away from the cell wall will be occurring. So this is the difference between plant cell and animal cells when they are being put in a hypotonic, hypertonic and a isotonic solution.